Dicot embryo development. See, before this embryo development discussion, we know fertilization. Mainly in angiosperms, two types of fertilizations. One is true fertilization and double fertilization. The double fertilization is also called triple fusion. True fertilization is also called a syngamy. One male gamete fused with egg cell to form diploid zygote. The diploid zygote develop into complete embryo in this syngamy fertilization. See, in angiosperms, mainly two types of embryos. One is dicot embryo, another one is monocot embryo development. Today we discussed here it is dicot embryo development. The dicot embryo development first identified in cruciferous families. So this embryo development is also called cruciferous type of dicot embryo development. In cruciferous species, that main species Capsella bursa. Capsella bursa belongs to dicot embryo, dicot plant. The Capsella bursa containing embryo, that development will discuss it today. It is the diagram of the dicot embryo development, Capsella bursa. See, after syngamy, syngamy means one male gamete fused with egg cell to form diploid zygote. The diploid zygote present in the micropylar region of the ovule or embryo sac. Initially, in Capsella bursa or cruciferous family, the diploid zygote participated in the first division is transfer division. By this transfer division, two cells are formed at the micropylar region. The two cells are in diploid conditions. See, one cell move towards or one cell towards micropylar region called CV cell or basal cell. One cell towards chalagal region, that cell is called terminal cell or also called CA cell. What is the first point here? It is diploid zygote participated in the first transfer division to form two cells. One cell present towards micropylar region. That micropylar region single cell is called basal cell or CV cell. And above this basal cell, one cell is present. It present towards chalagal region is called terminal cell. The terminal cell is also called CA cell. From these two cells, CA cell again participated in the longitudinal division. At the same time, CB cell also participated in the transfer division. See, here it is two different divisions by these two cells. CA cell participated in the longitudinal division to form two cells. At the same time, CB cell also participated in the transfer division to form two cells. At this stage, how many cells here? It is 4 cells are present. The 4 cells of embryo is called proembryo. Proembryo is a capital T-shaped structure. From this capital T-shaped embryo or proembryo, above 2 cells are developed from the terminal or CA cell, and below 2 cells are developed from the CB cell or basal cell. And CA cells, the 2 CA cells are again participated in the periclinal divisions to form 4 cells. The 4 cells are arranged in the 2 tiers. In each tier, 2 cells are present. It is lower tier is called I tier. Upper tier is called L1 tier. These 4 cells again participated in the mitotic division to form 8 cells. 8 cells again participated in the mitotic division to form 16 cells. The 16 cells are arranged in the 2 orals. Outer oral 8 cells, inner oral 8 cells. Outer oral 8 cells cannot participate in the further division. They take rest for the few moments. And inner oral 8 cells again participated in the mitotic division to form 16 cells. At this stage, Total 24 cells are present. The 24 cells are arranged in the three orals. They are arranged in a spheroidal structure. These 24 cells developed from only CA cell. Remind it. 24 cells developed from the CA cell. See, from these 24 cells, outer oral 8 cells act as a dermatogen initials. And the middle 8 cells act as a cortical or cortex initials. Inner oral 8 cells act as a vascular initials. See, dermatogen of shoot, cortex of shoot, vascular bundles or vascular region of shoot. These three orals of cells develop into complete shoot part from the embryo. See, these cells, outer oral 8 cells, they called dermatogen initial cells. The dermatogen initial cells participated in the periclinal or mitotic divisions to form more number of cells in the dermatogen region. At the same time, middle 8 cells called cortex initial cells. The middle cortex 8 initial cells also participated in the periclinal divisions. They develop into mass of cells. And the center oral or inner oral 8 cells are developed into vascular initials. They are also participating in the more number of cells by the mitotic division. See, by this division of mitotics, they develop into heart-shaped structure. The heart-shaped structure containing dermatogen, it develop into dermis of shoot, peribellum. The peribellum tissues are developed from the cortex initial cells. They are developed into cortex of shoot called peribellum, and plerome. A center region 8 cells are present. They called vascular initial cells. That vascular initial cells again participated in the mitotic division to form mass of cells. They called plerome. They develop into vascular region of the shoot. At this time, the mass of cells are arranged in a heart-shaped structure. That heart-shaped structure is developed by only CA cell, terminal cell. At the same time, during these division, CV cell also participated in the transfer division to form two cells. They are present one above the other. These two cells are again participated in more number of transfer divisions only. Only transfer division to form a chain-like structure of cells. The chain-like structure of cells are developed from the basal cell or CV cell. The chain-like structure containing more number of cells. See, from these cells, one cell is in large size. The large size cell act as a suspensor cell. The suspensor cell is large size. Mainly at the matured stage of embryo or during the development of embryo, the large size of suspensor cell act as a hustorium. Hustorium main function is collecting of food material from the endosperm. Already the developed embryo need nutrients, minerals. The needed nutrients, minerals are present in the endosperm. The endosperm supplies nutrients to the matured embryo or developed embryo. That nutrients are received by the hustorium. And opposite of these hustorium, in the suspensor cell, one cell is present. It present near the embryo. That cell is called hypophysis. Opposite of hustorium, hypophysis is present in the suspensor. It is the suspensor. Hypophysis is participated in the mitotic division to form 8 cells. That 8 cells are present in the 2 tiers. In each tier, 4 cells are present. See, lower tier 4 cells are developed into root cap, and upper tier 4 cells are developed into root apex. Means the complete root part is developed from the CB cell. and complete shoot part developed from the CA cell in the dicotyledons. And during the development from the CB cell, suspensor cell also developed by the basal cell or CB cell. In these suspensor cells, one is large in size, it acts as a haustorium. During this development, the heart-shaped embryo is turned into torpedo stage. It is the final stage. Torpedo stage embryo look like a shoe horse-like structure. It shows curved-like structures. The curved-like structure embryo containing only two major stages, two major parts. One is shoot meristem, like it is cotyledon, and root apex or root meristem. Two half parts are present in the matured embryo. When they developed embryo, they loses the suspensor, including haustorium. Finally, the matured embryo containing torpedo shape, it containing epicotyledon, hypocotyledon. Epicotyledon means upper portion of the dicot embryo is called epicotyledon. Epi means dorsal region or dorsal part of the embryo is called epicotyledon. In this epicotyledon, two cotyledons are present. Between these two cotyledons, meristem tissues are present. That meristem tissues are developed into shoot meristem. Mean complete shoot part is called epicotyledon, and hypocotyledon means complete root part. Hypocotyledon means it is the lower part of dicot embryo. It contains a root meristem. Finally, the dicot matured embryo containing only two major parts. One is epicotyledon, hypocotyledon. It is the dicot developed embryo development.